Good afternoon, everyone. Welcome to the second session. I have the privilege of introducing Dr. Pratibha Nadig, who has an illustrious career in her pharmacology field. Dr. Pratibha Nadig is MD in pharmacology from Karnataka Institute of Medical Sciences, Hubli. She has served various positions, including professor, head of the pharmacology, head of pharmacovigilance, research head, faculty medical education unit in Vaidehi Institute of Medical Sciences, also in natural remedies, KIMS SDM College of Dental Sciences. She is also a consultant family physician for over 10 years. In her 25 years as a clinical pharmacologist, in medical education, apart from teaching pharmacology, she has emphasized practice of safe and rational use of medications and also evidence-based medicines. She has conducted many sponsored clinical trials as principal investigator. So she will be the right person to address all your doubts and enlighten you about clinical trial protocol writing. She has trained many students in clinical research, from medical students, dental and pharmacy students. She has more than 35 national and international publications. She is president of South Zone of Global Pharmacovigilance Society, and she is also on the advisory board for Journal of Pharmacovigilance and Drug Research, and also co-chairman for Indian Medical Association, Karnataka State Branch. Currently, she is working as professor, head of the pharmacology at Dr. Chandrama Dayanand Sagar Institute of Medical Education, Bangalore. I welcome you, madam.
Thank you very much, Dr. Dhamle, for the kind introduction. Am I audible?
Yes, madam, you're audible.
Okay, thank you. So, thank you very much, Atrimed Pharmaceuticals, for this opportunity at the outset. My association with natural products has been quite long. My first venture into um, proving that natural products do play a vital role in health and disease started when I was a fresher after my MD pharmacology. I started experimenting with Philanthus neruri, root drug powder of Philanthus neruri. In fact, those days, that was in 1996 at Hubli, where there were not many regulations. So we just got the herbs and then we powdered them and then I got them encapsulated. And of course, with the ethics committee approval, we started a clinical trial. Not much was known or not much was expected whenever we tried to use natural products and even the new chemical entities also. So, over so many years, actually, we have grown with the ICH GCP guidelines because of many, mishaps during clinical trial or mishaps while experimenting with new products. Always there has been an update or amendment or regulatory requirement for making the product safe, more safe, more efficacious. Even if it is not efficacious, it's fine, but it should not cause any harm to the patient who has taken the medication as a clinical trial participant. So all this has come into existence, this proving that the medication works, as she has pointed out in my introduction. Uh, Evidence is required. Whenever we want to prove something, evidence is required. And there is a different hierarchy of evidence. If you just publish a case report, you say this product is working very well. That will be accepted, but with a pinch of salt and with doubts, because maybe this patient has responded, or there may be a placebo effect, things like that. If you publish a case series, like many cases you have treated, about 10 cases, 15 cases, I have tried this Philanthus neruri for diabetic patient and it has worked. Still, there is a query because they ask, now maybe you don't have a control group. Maybe that patient must have controlled on he must have exercised control on his diet, or he would have done his exercise. Likewise, there is always a doubt whenever we try to prove something, unless our scientific design or our methodology is very, very accurate and precise. So, this is what we have learned all these years. More so, you would have observed during the COVID pandemic time, lot of things came into forefront, including the drugs, repurposed drugs, the natural products, the diagnostics, devices, everything under the sun related to healthcare mushroomed like anything, and most of them fell flat because they did not have enough scientific data to prove their point. So therefore, writing a clinical trial protocol, conducting proper, well designed, well controlled studies with statistical backup is extremely important to prove that a product is working, or any of the device is working, or any intervention for that matter is working very well. Therefore, I assume most of you are interns. That is what I was told. I have tried to simplify as much as possible and to the best of my ability. There are some areas where you need to consult someone else, but what best possible and the way we design, I have tried to explain. Now, protocol is a very, very important document that will tell us how to go about experimentation. We don't have to prove that it is working, but the protocol should be so well designed that it should tell us whether it works or not. We must be open minded. The protocol should be totally, we use the word bias, it should be totally unbiased. So that is writing a protocol. I often give an example of, cooking, or riding a motorcycle, or riding a car. So when you write or when you explain the procedure, it should be in such a way that somebody who, everyone understands, first of all, everything, all the process, all the procedures should be meticulously defined. Secondly, if somebody wants to reproduce, it should bring about, I mean, they should be able to do it without any hassles. So that clarity should be there when we write a protocol. Many times, especially in the pharmaceutical industry, clinical trials are done for new interventions, maximum utilized by new chemical entities or new drugs. So, what happens is, if there is any ambiguity, people may try to utilize an ambiguous protocol to sell their product. Therefore, the regulatory, that means there is something missing. You may not know what it is, but it will be called as a protocol. So, this started happening, and therefore, the regulators and the ICMR and the Ayush, all these people have agreed upon following certain good clinical practice protocol. Good clinical practice is not as a clinical practice, but it is a practice which is followed when we do clinical research. So clinical trial is one aspect of clinical research. There are many other types of clinical research, but usually we use clinical trial and clinical research synonymously. But actually, clinical research, clinical trial is what we are referring to today. So most of you are going to apply for these projects, and you're going to see how you are going to design. So as we have seen in the morning sessions, and I think you have another session, any new drug development in the conventional, traditional way, a new drug development, whether it is new chemical entity or a herbal product or any other source, from any other source, it goes through different phases. And like drug discovery, first we come out, we identify few promising molecules, then they undergo extensive preclinical studies, as we have discussed extensively in the morning, where animal studies are done, and some studies are undertaken in the laboratory. You can stop me at any time, but I prefer to take the questions in the end. But if it is so urgent, you can interrupt. So preclinical studies are animal studies, and then after we are convinced with the preclinical studies, then the product come into clinical trials, and then they are marketed for use. This is a broad, I'm sure all these speakers will go through this, because clinical trials form an integral part, because that is where we are going to prove that the medication or any intervention is working, and if it is working, to what an extent, and all those details. So basically, a clinical trial is a biomedical or health interventional research in human beings on a predefined and an approved protocol. And we must understand that it is not used to treat a patient. We are observing here. Patient may get benefit, may not get benefit. We must go with an open mind as we design the protocol. We must go with an open mind to precisely identify whether it is going to work or it's not going to work. Therefore, clinical trial is an experiment. if you have seen clinical research designs, this is an experimental study. It is an interventional study. It is an experimental. Unlike case report, case series, those are all descriptive, cohort, case control, all those are descriptive. This is an experimental study and it is not a therapy, but patients are expected to improve based on our preclinical studies. But what is the extent of improvement that you have to observe? So it is a written procedure. Protocol is a written procedure where everything is defined. First of all, we understand why clinical trials. We all know that for new drugs, combinations of medicines, new use of existing medications. We all know that during COVID, we had a lot of such repurposed drugs. They were used for something else, hydroxychloroquine, and we brought it for, which was used in malaria. We brought it for COVID, medical devices, pulse oximeter, surgical procedures, diagnostic kits, all these go through clinical trials. Good clinical practice has to be followed when we conduct clinical trials because it is the standard for design, conduct. Design means protocol, what we are doing now, for the conduct by the investigator and for performance overall. That means entire management. We shall see how, if you go by the sponsored trial, now what you people are going to do is in a small population, you are going to evaluate a product if you are undertaking clinical trial. I am speaking from broader sense. Yours could be one of the center. If Atrimed decides to conduct multicentric trial, yours may be one of the centers, but good clinical practice is followed for the clinical trial of that product across all centers, so it is a standard for design, that is protocol, preparation of protocol and all other documents, for conduct, for performance, for monitoring, assuring that it is of high quality, for auditing, for recording or documentation, analysis and reporting. So you understand there are so many steps for a pharmaceutical company to bring out a product. At one center also there are so many tasks to be undertaken. As much as possible we need to adhere to that, because whatever data we get out of that, that will be credible and accurate, and it protects the patient's rights, safety and well-being. So we have quality as well as we are ethical. So a protocol follows good clinical practice, and good clinical practice is not only for protocol but it is for entire system of uh, of conducting the evaluation on humans. So that is all covered under good clinical practice. We have several guidelines. One is International Conference on Harmonization, ICH GCP. Then we have Indian GCP. ICMR also has come out with guidelines which is similar to Indian GCP. So we all know that every drug conventionally, I'm talking about new chemical entity. When a new molecule is synthesized and we want to explore, then after preclinical, it undergoes different phases. Phase one study, step by step, because at every step we need to know we need to have confidence that it is safe and it is efficacious. Efficacy will come clearly. We get a picture of efficacy as we finish, but here we should be very well assured. We should be assured that it is safe and we determine the dose. So after preclinical testing, the regulatory bodies will approve, and phase one is conducted on healthy human volunteers. For this also we need a protocol, but it depends on the objectives. So here we are. I will come to that as I run through the headings. So we explore the safety and dosing of the drug, and we decide the what could be the tolerable doses in humans. So this will be the first human study. In fact, now we have phase 0 studies done in very small percentage of population, maybe 5 to 10, because the manufacturer, the pharma sponsor wants to see whether it gets absorbed. Let's say it's meant for oral use, whether it gets absorbed and what is a rough pharmacokinetic profile of the drug. Pharmacokinetics means, as you all know, whether it gets absorbed and how much gets metabolized and all that. So they will have some idea, because a lot of investment goes in design and conduct of these clinical trials. So phase one is exploring this. And after that, once we have confidence, we go into evaluating the effectiveness in disease patients. In patients, this is in healthy human volunteers. This is in patients. Then more number of patients in phase 3. As we come to phase 3, we do in multiple centers. Then again, we apply for approval to market the product, and FDA or the CDSCO, Indian Regulatory Authority, Central Drug Standards Control Organization. This is US FDA. So then they give approval for marketing, but closely they monitor, they review, and then they give approval for marketing, where we evaluate in larger number of patients who are prescribed. Until here it's trial, here it is prescribed but under stringent observation. So this is how step by step it goes. And for a totally new chemical entity such as, let's say, atorvastatin, which we use as hypolipidemic, or maybe glibenclamide, which was used in for diabetes, is used for diabetes, for such products it takes at least 10 to 15 years for the entire new molecule to come into market, and now it has crossed $1 billion. So that much is an investment for a new, because safety is questionable, because we don't know completely how it's going to interact with. Any questions so far? Now, one thing I will clarify before you ask questions: for the herbal medications, they don't come under strictly these CDSCO or trials. Ayush has made some regulatory guidelines, but CDSCO also has made guidelines for isolated molecules. Let's say curcumin from curcuma longa, that has to undergo a detailed similar to this. Of course, curcumin is there in the market in different version, but something like that. If you bring out new, if it's a chemical, then it has to undergo. Otherwise, for extracts there are different guidelines, and for crude drug powder there are different guidelines. CDSO doesn't the same CDSO which approves a new chemical entity will not go through. Anyway, that is not required. Now let us get into the proper, these are all the stakeholders in clinical trial who are all involved. So that is by the requirement for the sponsor. So if you are bringing out a product and you want to study in any center, then it will be the hospital. Let's say our hospital is doing here. This sponsor comes, brings out the product. He may give it to clinical research organization. Through them, it goes to clinical trial site. Then study participant is one of the stakeholder, regulatory authorities, and overall IRB IEC is the most important stakeholder, which actually is concerned about protecting the patient's rights, safety, well-being, which will oversee. And it's much more, in fact, they sometimes place it higher than regulatory authorities, because we are in close contact with the clinical trial team as well as the study participants. So these are the stakeholders. So then we start with actually how a clinical trial protocol is designed. I thought this background would be required for us to understand the necessity of writing a protocol and how much is involved. Now, if you don't go through CDSCO, any new formulation, Ayurvedic, anything, you are, Atrimed is making an attempt to orient all of you to understand that it has to undergo a scientific validation, because we know it has been used since ages, and we know there is human usage, and we are going to use exactly in the same method as described in Ayurveda. But in order to justify as per the parameters or biochemical investigations that are described in the current medical literature, you need to prove it, and therefore we are going to do. So which phase to follow? You can follow the regulations, but at least one cohort, if you have the preclinical toxicity data and preclinical evaluation data in animal studies, with that we can enter into clinical trial protocol. Any doubts so far? All right. So writing a clinical trial. Yes. Okay. Somebody has a question. Can you please unmute and ask?
Yeah, hello, ma'am.
Yeah, please tell.
I'm Dr. Sumaya. So,
Sumaya,
ma'am,
yeah.
I want to put across a question. Uh, so basically, for example, if a doctor who is not associated with any institution, who doesn't have a hospital, but who has a clinic and would like to do the trials of maybe certain therapies, maybe some basti therapy, or maybe any dhara, any of these therapies and action of that therapy, is there a possibility where one can do a research and actually submit a study?
Yes,
And will that be considered authentic? How?
yes.
And how, ma'am?
Yeah, you can do that study. You must get approval by the independent ethics committee. There will
Okay,
be
and who?
independent ethics committee which are not affiliated to any institutions. There are.
Okay, ma'am.
So get an approval from there. And now it says 50 kilometers. The committee office may be located 50 anywhere in the 50 kilometers radius is open. Yeah, they can approve your protocol. There are some institutional ethics committees who are willing to give approval if it is there in their SOPs, which is approved by the CDSU.
Okay, CDSU.
CDS, our ethics committee, not you, not the investigator, not the doctor. If
Yes,
usually no, the ethics committees are will get accredited. It should be the ethics committee must be recognized by CDSU, Government of India. Such ethics committees are existing. If you approach them, they are open and they are willing to approve, and they are actually made for approving protocols from independent.
Yeah, ma'am. The other question I have is, suppose once you get approval from the committee, so the drug and such things, suppose these are pre-approved drugs, but just want to know the efficacy of the therapy. Now, this is something we can make a study, a proper hypothesis, a clinical trial, pre and post clinical trial tests, and we can proceed. Is that what I understand?
Sure. Yeah. In the meanwhile, we were discussing about the contents of the protocol. Extremely sorry for this internet problem. So there are a lot of headings under which a protocol is designed, and it is essential because as I mentioned, every detail has to be documented, and that is a source document based on which we are going to conduct the clinical trials. So that is about the contents. Now the first one, you will soon see the slides. The first page will be on general information, where we need to give the title and we talk about the date and the details of persons involved. Protocol, as I told, is a very, very critical document. And when you write also, because it's a reference for you also, so you write your name as an investigator or your senior or your guide, whoever is guiding you. But in terms of pharma industry, they write the name of the sponsor, the CRO, and they might have employed a medical monitor, and who is the investigator, which is the laboratory where we are going to send the samples for investigations. All those details they are mentioned on the protocol, because as I said, accountability is important. Who wrote this protocol? Who is the manufacturer? Because as I told you, who are all the people who are seeing the protocol? It is handled by the government, it's handled by ethics committee, by the investigator, by the monitor, and everyone. Okay, I'll come back to the contents. Without wasting much of time, the first thing which we start writing is the background information. So can anybody tell me, like when I want to, the background information is like telling a story about this product. We have brought this product and we are telling the investigator or if I'm a sponsor, I have a product, I will go and approach an investigator, or if I'm an investigator doctor, I'll write a protocol and I have to submit that before the ethics committee to get permission to conduct a clinical trial. So my protocol will be written like this. The first thing will be the background information. That will be an information about the medication so far, whatever is known, right? Whatever is known about it. So as I told you, there are different phases for new chemical entities, and let's assume you are investigating your Ayurvedic product and you want to write a protocol. So you must convince the ethics committee or whoever is reviewing that your product so far has shown some promising results, and now I would like to evaluate in the on humans, and what is known about this product so far. I will build up a story, and that story will include certain things. So let us say we want to investigate Nisha Maliki. Nisha Maliki is a product which I had heard for diabetes, right? So Nisha Maliki is a very wonderful product which is used in type 2 diabetes, and I want to write the background information. So can somebody tell me what information would you like to give to the ethics committee to get approval and convince them that it is justified to conduct trial on this and to get more data, so that people will accept your product. I mean, people, it will give some relevant, it will show whether it is working and how much it is working. So how will you convince that this trial is necessary, and how will you convince so far, or what data would you have so far on this product? Anybody? We are conducting the trial on human beings. Let us say it's a phase 3 trial. Let me call it as a phase 3 trial. Or so what information would you like to write, or what information you have with you, or you would have searched some literature, you would have seen some publication. So what information would you like to give? Anybody wants to volunteer? I think we have to get participants to speak so that it's nice to discuss. Anybody would like to, one point you can tell about this, about a product you want to investigate. It's a known Ayurvedic product. Anybody wants to say? Please volunteer, even if it's wrong, doesn't matter. Please, yeah, please feel free to answer also, contribute.
Yes.
Anybody? Something you have to tell. We have heard in the morning about preclinical studies, isn't it? Would you like to, supposing it has undergone a preclinical animal study, would you like to put it in the background information for a clinical trial protocol? Yes or no? Dr. Sumia, would you like to answer?
Ma'am, I couldn't hear you for a while. I was disconnected. Could you please?
Okay. So this protocol writing starts from writing the background.
Yes, ma'am.
Slides are visible?
Yes, ma'am.
Not yet?
No, visible.
They're visible.
Yes.
Visible. So I have, please go to the slide on background, Mr. Prithvi, background. So here I have shown a story. So we tell a story about the product so far, what has happened. And then we convince. So either Atrimed will come and tell your institute, or you as a doctor, please do a study. Now they would have asked some Atrimed products.
Yes, ma'am.
So when you write a you will take it to ethics committee to get approval.
Yes,
so ethics committee will, that protocol background should be convincing enough to the ethics committee that this product has some stuff, either in terms of both in safety and efficacy. So what data will you give? What information will you write in the background about any product? Like I told Nisha, is type 2 diabetes. It's a product, or glimepiride is used. So what information you think we could write under this heading?
Ma'am, the anti-diabetic effect of Nishamiliki, if it is already there in the literatures before or any studies done.
Yes. Yes, very good. Exactly. That's what I wanted. So whatever is there in literature, you write that with references, and also if you have studied on animals, you mentioned that. If you have done toxicity, preclinical toxicity, you mentioned whatever is known about this product, which will add value to safety and efficacy, that goes in the background. And then we say, yes, so much is known. Now we would like to investigate further. So next slide, please. So it will talk about name and description of your product, non-clinical studies, known risks and benefits. If you should be very upfront. So you can say that some patients have shown adverse reactions. Maybe we want to investigate further and describe why I should use it orally. It has shown in animals and it has been observed. And morning madam was telling bioavailability data. If you have, you can mention that. You can say we did something on Syzygium cumini also. Recently we had done one study. So ellagic acid was there in the blood. So like that, you give justification as much as possible in a convincing way. So that should be written, and say that this will adhere to GCP and on whom we are studying. And then in the end, actually, this is as per ICH GCP I have taken. But now you can say, and therefore we would like to investigate further in humans and get the data and see whether it could be popularized. Next slide, please. So now we are going to trial. Then after that, background with references. If you see that it ends in references. Now we go to trial objectives and purpose. Is that slide visible? Yeah. So
We are
under
in the
this,
trial design stage.
Go back, please. Go back. Go to the background information. Then we have trial objectives and purpose. Slide number 13. Trial objectives and purpose. Is it visible? Okay. So we now talk about next. After we write the background, we go to trial objectives and purpose. So you must be very clear. You want to evaluate Nisha Maliki, detailed description of the objectives and purpose of the trial. So here we are going to talk about what would be our primary objective, what would we divide them into primary and secondary objectives. So if you see, I have given one example. to evaluate the efficacy of Nishamalaki in comparison with metformin in uncomplicated type 2 diabetes mellitus patients. So during this slide adjustment, one thing we have missed is the title. So this is the title of this example which I am giving is, it's a double-blind standard controlled trial of clinical trial of Nishamalaki in comparison with metformin in uncomplicated type 2 diabetes. That was a title. Then now we are talking about, that's the title, that's over. Then we wrote the background. Now we are writing the objectives. So our objective is to evaluate the efficacy. The first one is efficacy of Nishamalaki in comparison with metformin in uncomplicated type 2 diabetes mellitus. This is how we need to state. So protocol is all about writing using the appropriate words. If you see, we have written to evaluate. What are we evaluating? Efficacy in comparison with metformin in uncomplicated type 2 diabetes mellitus patients. Then one more objective I want to put is to evaluate the adverse effect profile of Nishamalaki in comparison with metformin in uncomplicated type 2 diabetes mellitus patients. So this is our primary objective, secondary objective, and objectives must be very smart. Smart, they say, use the adjective to describe how to write. Our objective should be very specific. Are we specific? Yes, we are evaluating efficacy. And it should be measurable. Can we measure the efficacy? Yes, we can measure. How do we measure the efficacy in diabetes? We check for the fasting blood glucose and or HbA1c. And is it achievable? Yes, it is achievable. Is it reliable? Yes, these are reliable because we know most of the diabetic agents, anti-diabetic agents will be done. Is it timely? Yes, it's timely. If we add some more, when administered over a period of 3 months, we can write, or we can leave it there. But basically it should be very specific, it should be measurable, it should be achievable, reliable, timely. Stating your objective is very, very important because the entire protocol, the procedure depends on what you have stated as objective. I will again come back to objective for every step when we write the protocol. Next slide, please. Now define what are the end points. What are the end points? What are the end points you must define? And now if I want to the efficacy, then what would be my fasting blood glucose? Initially, let's say the mean, if the mean blood glucose was, let us say 200 milligrams when after treating the patient, after patients with either Nishamalaki, one group maybe they showed from 200 the fasting came to 100, the other one showed to 140. So what is this mean change? I'm talking about the mean, all the people in the end. So what we want to see, I'm not defining now, must say we are going to measure change in the fasting blood glucose or change in the HbA1c. So based on the primary objectives, the endpoints are decided. That is why I have put objectives and corresponding endpoints. I hope it is clear to all of you. So based on the primary objective, what is the primary objective was to evaluate in the fasting blood glucose. What is the change in the HbA? The objective was adverse effect profile. So endpoint would be incidence of adverse clinical events compared. So endpoint would be the intervention and the control. So what is the intervention? Nishamalaki. And we are observing on a specific way and will determine if effective or not. Now we is here. Outcome usually refers to the measured. What happened to mean HbA1c before after? Endpoint is later. That means that from baseline, how much change was happening in a metformin group versus how much change was happening in the Nishamalaki group. So we are going to compare that using proper statistical analysis. Now we talk about trial design, description of the design of the trial to be conducted. What is randomization? What is blinding? What is a control? And what is a crossover? Go to the next slide, please. So randomization implies there is no rule or predictability for allocating the eligible subjects to treatment and control group. That means we don't want to say this person should go to A group, this person should go to B group. No, there is a method wherein we randomly allocate because it will avoid bias. Now, what is a bias? In any experimentation, scientific experiments, we should not be having any preconceived idea. For example, I am an allopath. I will say always metformin is better. So I will start putting, let's say, mild to moderate. It works in mild to moderate diabetes. I will allocate all mild to moderate diabetics in one group, metformin group, all moderate to severe in Nisha Malki group. Somewhere I want to show that my metformin is better than Nisha Malki. No, it should not be like that. We should be open. That preconceived idea is called, preconceived notion is called as the bias. So avoid that because it should be totally open. We must be open for positive or negative results, and therefore this randomization ensures that they are allocated to both the groups. Who is allocated? Whoever is eligible. I will tell you what is eligibility. You know that. So first we will define who are eligible. That comes later, inclusion, exclusion criteria. Then that person should have equal chance of going to either Nisha Malki group or metformin group. So there is a method, computerized method for the, so that takes a longer time. Right now you understand that we generate a randomization schedule. When we write a protocol, there is a way, if you go through the Google and ask them to generate a randomization table. So first patient, let's say, go to A group, second person goes to B group, third person goes to B group again, fourth comes to A, fifth comes to A, sixth comes to A, suddenly seven goes. So like that, to avoid bias, there are certain randomization schedules which are there on the internet. We go through that and we generate a computer generated table, and then we give it to the investigator. I'm talking, if I am a sponsor, I want to do a trial with you as a doctor, I will come and tell you that this patient should go to A group. He doesn't know what is A, what is B. I would have covered, I would have blinded it. So he will say go to A, go to B. Or I will say, if it is not blinded also, I will tell randomization can still be done and we need not blind also. Just randomization. Follow the schedule. So I will tell him, so he will start first patient, okay, he's given metformin. Second patient is given Nisha Malki. Like that, based on the table, based on the which we give it to them. Next slide, please. So randomization in clinical trial, this is how it works. So if you see the first one, details of all the patients who are taking part in the trial is put on the computer, and computer generates a schedule. And now this here, what I'm talking is, there's something called as the IVRS. It's called Interactive Voice Recording System. In a multicentric trial, what we do is the investigator feeds in the data on the computer, and then immediately it says, okay, allocate him to the software. It would be programmed in such a way that it will immediately tell you this patient has got this much of fasting glucose, this is the thing, left in normal, everything in normal, and he's the first person. Okay, treatment A, it says, then you allocate. So ultimately, if you see the set, that is IVRS. Otherwise, you are given a sheet. First patient should go to metformin, second should go to Nisha Malki, like that sheets are, like that schedule is given. So if you see in the end, the table would have generated, see the third part of this picture says, in A and B, equally all types of patients are distributed. So any variation that happens due to time or season, exercise, whatever is going to affect, will affect both the groups equally. So therefore, there is no effect of any confounding. Some amount will be there, but nothing else will affect. Anything will affect both the groups equally. That is the purpose of randomization. Go to the next slide, please. Then we come to what is blinding. Why blinding should be done? Again, to avoid bias. Supposing the doctor knows, as I told you earlier, doctor knows that this patient, I know he will respond very well because I have put him on metformin. I knew it will work. Or an Ayurvedic physician may say, no, Nisha Malki, always I have seen it works better. So let's make the investigator blind. He will not know which treatment he is giving to which patient. So we will make the look-alike tablets, so that he doesn't know. We only label the container. So where the one person is not aware, that means sometimes the investigator knows, but the participant is not aware, because the patient may say, oh, I'm taking Ayurvedic product, so I'm good, I'm getting better. Or the some patients may be for allopathic, they may say, I'm better with metformin. So that bias also we want to avoid. So if the participant is not aware, but only participant is not aware, it's called as the single blind study. And where participant as well as the investigator is not aware, it's called double double-blind study, and where the participant, the investigator, as well as the monitor is not known. It doesn't know what it is. Only the statistician or the third party has the code as to what is A, what is B. Then it is called as a triple blind. So more the blinding, more is the more scientific it becomes, because we avoid bias at every step. One step we avoided in randomization, the other step we avoided in blinding. But one thing we should be sure, we should be careful. Sometimes there may be adverse effects, very severe adverse effects. The patient may experience hospital emergency may come, he may go into some complication. At that time, there should be a procedure on how to unblind and what the treatment should be given. That also must be mentioned in the protocol. So in clinical trial design, you will say it is a double blind or triple blind, randomized, and then please go to the next slide. Parallel versus crossover. Can you see that slide?
Yes, ma'am.
Okay, so the third one is whether it is parallel or crossover. Sometimes we give the medication, let's say give medication A, and for 3 months, medication B to another group for 3 months, and then analyze the results. So that is parallel study. Sometimes we give a washout and then crisscross. So what is the need for the same patient to take Mishamaliki also, give a washout, take metformin also? Anybody from the audience? Anybody can tell me what may be the added advantage of crossover? Crossover is time consuming, it is expensive, and for the patient also, but it gives better results. That is what I feel. Anybody from the audience? Okay. So what happens is, in parallel, we are seeing what changes happen before and after, and then compare with these two groups. In crossover, the same patients will be exposed or will be taking one medication, then wash out, then second medication, so that there is still further bias is avoided, because here the whatever genetic or epigenetic or whatever his body reacts to that drug in any form, maybe kinetics or dynamics, we get the results for both the medications in the same patient. How he responded to hemoglobin A, because although we have done randomization, we have done so many criteria, we have done blinding, still an individual makeup may influence the response to treatment. Individual makeup, he may respond in a different way to Nishamalki, and he may respond in a different way to metformin. For that, we have taken care. We have taken big sample size. Everything is okay. But still, to be doubly sure, we will expose the same patient to both the medications. So that is it. Go to the next slide, please. Yeah. Can you see this?
Yes.
Controls in clinical trial. Yeah. So we can use either placebo, which is inert substance. We can compare Nishamalki in diabetes without, and another group can be just a placebo, no treatment. Or you can use a standard. Okay. So what is the reason why we can we use placebo in clinical trial always? Can we use anybody? I think Padmaja, if you're online, you answer this. Can we use placebo? Placebo is an inert substance, just to blind or avoid bias. Yeah, Padmaja, can you?
Yes, ma'am. We can use placebo control, placebo as one of the groups, ma'am. The trial to check for the safety and efficacy both.
Both we can do. Yeah, provided it is
Yeah.
mild diabetes, right? Yeah, provided it is mild. Yeah, because what happens is, if it is moderate to severe, then he may be put to risk. So therefore, placebos are used only when the standards are not available. For moderate to mild cases, okay, we can still observe, and we can see without treatment what happens. But for a severe condition, moderate to severe, we cannot risk the patient. So always we should use standard, unless you don't have a standard. But even then, we must have all the, if you are saying, you must be very careful, and you must have all the measures to manage the emergencies whenever we don't use. Okay,
Yes,
go to the next slide.
one more point, ma'am.
Yeah.
Whenever there is, as you told, standard available to do the comparison with respect to efficacy and safety, in most of the cases, standard will be the one group, ma'am. Placebo
Yes.
will not be included in most of the clinical
Yes.
trials. Most of the
Very good.
times, we will do a comparison kind of studies only.
With the standard, yes.
Yes,
Yes. Very good. That's exactly. We always use standard wherever it is available. If it is not available, and yeah, we will use the mostly we use standard only. So because of this issue, isn't it? If there is any emergency, then it will be very difficult. So
Yes,
that
ma'am.
is it.
Ethics also is an issue there.
Ethics also is an issue. Yes, ethics also is an issue because we are withholding the treatment. Correct.
Yes,
We are
ma'am.
withholding treatment when it is available. Very good. So next slide will be trial design. So now continuation of trial design. We have talked about what is the we quoted as it is randomized, double blind or triple blind, placebo, standard controlled trial, clinical trial of Nisha Maliki in type 2 diabetic patients, uncomplicated type 2 diabetic patients. We should state that design. And then we have talked about the objectives, we have talked about the endpoints. Now we are continuing with trial. Anybody has a question, you can unmute and ask. So now we talk about treatment and dosage. You describe the protocol now starts writing about what are the treatments. So treatment A, one please unmute if you are not. Yeah, if we are using the metformin, what is a dose? How many times you have to use? If you are using Nishamalki, you explain that and how you are going to use, how many times on empty stomach, or all those details should be mentioned in the protocol so that whoever is using will understand duration and follow up. We give them, then we call them for every 15 days. And when they come for follow-up, what do you do? Do you check the adverse drug reaction? You check for compliance, you check all those details should be mentioned. And then you talk about discontinuation criteria. When will you discontinue? Will you continue? He'll come for follow-up. He says, Doctor, ನಂಗೆ ತುಂಬಾ vomiting ಆಗ್ತಾ ಇದೆ, ಮೈಯೆಲ್ಲ ತುರ್ಕೆ ಬರ್ತಾ ಇದೆ, and all that. Would you like to continue? No. If the patient has got moderate to severe adverse events, then you will like to discontinue. Or if he's not showing any improvement. So all this you should mention in your protocol. You should imagine, you should think about all this. What could happen? Because when you're a clinician, you know, you would have seen the patients. What could happen? So mention that. And then you talk about drug accountability. There's a special procedure for drug accountability. There are logs mentioned that the company will tell you, because whenever you dispense, no, every 15 days you call the patient, you give him one bottle which contains 15 tablets, let's say, for once a day administration. And then you ask him to come back and then get those medications and you check for compliance. Also, the drug accountability means when you're dispensing from the pharmacy, the pharmacist will write down, he will account. I received so many boxes or so many containers from the sponsor, and how I have dispensed, how many, he should give that account to the sponsor or the CRO, which is actually monitoring the study. And then procedures for breaking the course. As I told you earlier, sometimes the patient goes in for severe adverse event, or he will not show any improvement, his diabetes may worsen in this example, or whatever be the condition. At that point of time, you need to break the code and see what is taking, because you have to treat him. So what is the procedure and how do you manage? So next slide, please. Now we are going to talk about how do you select your patients depending on your disease, depending on your product, for whom it is meant, which age group it is meant, which condition, is it new or old. So in our study, we said randomized double blind metformin control trial, clinical trial of Nisha Maliki. So just for gives few examples, we say who are to be included: type 2 diabetes with FBG more than 126, because that is the criteria. Age group, you can change it. I have just put 20 to 40. It is more because we are seeing early diabetes now. You can change it. And newly diagnosed or already. So you, according to where you want to study this product on, which specific group, you can include that and exclude whichever is going to interfere in your observations. One thing you must remember: opposite of inclusion criteria will never be an exclusion criteria. Let's say if I write type 2 diabetes mellitus, then you cannot write type 1 diabetes here. No, it is for the same patient, something will come on the way. We said uncomplicated. So here you can say if serum creatinine is more, or if there's neuropathy, if there's retinopathy, if the liver, if there are any other comorbidities such as renal failure. So any such thing which is likely to influence the treatment with the study, which is likely to influence the study of Lishamalki as well as metformin, standard control. Anything which influences the outcome or the performance of your study drug or the standard drug, all those will come under exclusion. Very old patients we don't want. Creatinine, renal failure we don't want. Neuropathy we don't. So you exclude those patients and you define very clearly. And your inclusion criteria should be on par with the standard criteria, such as Indian Diabetic Association for Diabetes or American Diabetes Association global standards. So refer that and give the criteria according to that. So that is how you will define your inclusion and exclusion. And again, you can mention when to withdraw the trial subject from the study. That can come under this heading also. So that is about inclusion and exclusion criteria. Inclusion criteria, actually some of the new chemical entities give about 20, sometimes I've seen when we reviewed the protocols 20-25 inclusion, because they want to be very, very, very specific. But normally when people like I and you do academic research or some natural products which are safer, and we want to define some criteria, broad criteria, no? So that time you need not make it very rigid inclusion criteria, but definitely do not try to see what can influence. Those are called confounding factors, and put them under exclusion criteria. Exclusion criteria will be something that can interfere with your results. So that is about it. Go to the next slide, please. Then treatment of the subjects. Treatment of the subject. So treatment, again mention the names of the medications, and here we talk about rescue medication, and also under exclusion also we write what he should not be taking. But here now we are writing, supposing the patient goes in for severe diabetes, so what should be done? Can I give him insulin? Yes. Which insulin you will give, how much you will give, when to give, and how to monitor. And still if it is not controlled, withdraw the patients. Those details should come under here, apart from the treatment with the medication. Next slide, please. Assessment of efficacy. Is that seen? Assessment of efficacy.
Yes, ma'am.
Okay, so you define what are your efficacy parameters. So we talked about endpoints as well as outcomes. Endpoints is the change in HbA1c, and outcome was HbA1c. Outcome parameters are fasting blood glucose, HbA1c, lipid profile, or many more things, which quality of life. All these could be your outcome. Endpoints would be change in quality of life score, change in HbA1c, mean HbA1c, change in fasting blood glucose. So that you mention, and then you should also describe methods and timing for assessing. HbA1c will be recorded before the commencement of the trial and 3 months after the medication is given. And if you're extending, if your trial is 6 months, so they may say every 3 months. If fasting blood glucose, every month I would like to measure fasting blood glucose till the end of the study. And then you should mention which method, glucose oxidase method, HbA1c, which method. So these things you have to mention in the, you have to describe every detail. As I told you, so they will question you, which method you're following, is that method valid, is that kit very much is it standard kit you are using? So all this, because that will define your endpoint. Your whole protocol success depends upon what parameters you have chosen for comparison. So if that is not accurate, and if your methodology is, somebody will use one method, somebody will use Folin-Wu method, somebody will use glucose. So there are different methods. So you must define the methods that you are using. So next slide, please. Then assessment of safety. Again here, how do you assess the safety? You may record the serious adverse events like hospitalization. There are 5 serious adverse events. You can go through WHO criteria. Adverse event is any event which is seen during a clinical trial. You may feel, no, Nishamalki is not known to produce. No, we are not concerned. Whatever is seen in that patient during a clinical trial, even if it's false or an accident or whatever, any adverse event which is observed during a clinical trial must be recorded, and that is called as an adverse event. If you later we do what is called causality assessment to see whether it can be due to the drug metformin or Nishamalki, and here if it is blinded, we don't know anything whether he is on Nishamalki or whether he is on metformin, we are not aware. Therefore you must call them as adverse event. Serious adverse event is when there is hospitalization, when there is life-threatening condition or death or congenital anomaly or disability. All these 5 come under serious adverse events as per WHO. So you specify in your protocol, whenever these patients are put, the following are considered as serious adverse events. And adverse event is any event that can happen when the patient is on clinical trial, whether it is due to the medication or not. You must mention, give these definitions, write them in the protocol. And then you must say how will you record, how will you assess, and one more thing is how will you manage. That also you have to write. I have missed it out. Please write. This is as per GCP, but I would say you write how will you manage. If there's any adverse event there, the patient will be called back immediately and he will be managed as per the standard protocols. If it is severe, then the code will be broken, and depending on whatever medication he's taking, according to that he will be analyzed. So these details should come. So whenever you write any step in the protocol, you must think that he's your patient and he is undergoing. You imagine every step that he's going through, and then you will be able to write. And I will tell you very clearly that I have written many protocols. So when you start writing, you should remember practice. You should remember your patient that you are treating their patient. So you don't know, you are blinded about the medication, but you are supposed to manage him any kind of an adverse reaction. It's not a reaction. So be careful. Next slide, please. Then there is statistics. I cannot go too much in detail, but in the protocol, what is most important is you must calculate the sample size. This slide is clear? Is it visible? Statistics?
Yes, ma'am.
Okay, so what is most important is you talk about the sample size calculation. How did you arrive at? There is a formula to calculate the sample size based on previous literature and the estimated difference between the two medications, if it is known in the previous trials, or if you know any publication. Nishamalki showed improvement in about 90% of the patients. So like that, based on that, you can calculate. And two more parameters are error. How much of error is permissible? You can permit usually 5% error we can permit. 5% means there is 5% of chance that the improvement, that means one drug over the other, is by chance. It's not a true. That means 95% of the times we are correct when we say this drug is better than this. Either way. So sample size depends on all this, and level of significance, that means whether the difference observed. Let's say you get the change in fasting blood glucose 50% in metformin and only 20% in Nishamalki. So this difference, how important it is, that will be decided on based on some statistical tests which you would have studied. So I would suggest that you can go to a statistician to calculate appropriate sample size, and based on the literature, and show him the difference what has been observed in the published literature, or based on the sponsors data. Based on that you can decide, and decide the level of significance, that is called probability of occurrence of by chance, p-value, and which statistical test you are going to apply. If you are measuring before and after, there is one test. If you are doing it in the same group of patients, there is one type of test. If you are measuring repeated measures, that's called analysis of variance. So like that, there are different statistical tests. So that is a totally a different, a new, I mean, it's a very vast topic, so we cannot cover completely, but I can just orient you to this fact that statistics is extremely important step, which should not be taken in the end of the study. No, while planning a protocol, you must plan that this is how it's going to be done. This is going to be the sample size. These are the groups, and I'm going to give allocate the patients to this group. I will have so many people. This another group, I'll have so many people, and then I will see the change in, let's say, glycosylated hemoglobin from baseline to the end in one group compared to the other group. And I'm going to apply maybe unpaired students t-test. My p-value will be less than 0. If my p-value is less than 0.05, then only it is significant. All this will be written there. So that we should write. And then of course, along with statistics, then we should talk about missing data, unused data, spurious data. While you do statistical analysis, the bigger companies and multinationals, they have to give, that is called clinical data management. And now it's become a big thing that, I mean, how do you account for missing data, unused and spurious? Spurious, you can make out some blood pressure of 10 patients. Continuously 120/80, 120/80. This is all spurious data. It's not a true data. So whether you will take it for analysis or you will not. So statistical analysis is the final thing which will give you details. Go to the next slide, please. Then we have one more thing, direct access to source data or documents. This is when a sponsored trial is there. So investigator or institution will permit, you must write in the protocol, that they will permit for audits or by the regulatory agency or by the sponsor or by the IRBs or regulatory inspections. Next slide, please. Then ethics, very important in clinical trial, most important as science and quality is the ethics. So here you must write, the protocol will state that the study will commence after taking the institutional ethics committee approval. And at that time, the thing is you must go to the ethics committee and get it approved and explain, and ethics committee will ask various questions. Mainly they are concerned with the patient protection, whoever is participant, how safe it is, how are you managing adverse reaction, and how will you collect the data? Are you going to collect authentic data, or are you going to cook up the data? Do you have enough patients to study this? Or if you're studying in some other center, now Dr. Javed was asking me, Sumaya was asking me whether I can do it in my center. You can do it, but in given period of 1 year, and if you have a sample size of, let's say, 300, will you be able to do? Or let's say 100, will you get those patients? Can you follow? Is the study requiring follow-up of 100 patients for 3 months? Then you will have to see all that. You have to work out. So ethics committee will see all this, and then they will understand whether you are capable of doing it or not. And because the study should not be done just for study purpose, it should be giving some valuable output which can be used for patients. So data which you collect, and if the company markets that product, it should not be a fake product based on fake data. We have seen that in COVID times also, isn't it? So the remdesivir, how people manipulated the data, and it was published in very high-end journals like New England Journal of Medicine, and they had to withdraw. So these things should be taken care, and that will be seen by the ethics committee. And you must present before ethics committee, and you must take voluntary informed consent from the patients, because the patient should be aware that he may be exposed to either this drug or that drug if it is a randomized control trial. And then if he's convinced, only then he will sign. There should be no coercion. All this should be taken care, and these form the ethical standards of conducting a clinical trial, and that should be mentioned, written in the protocol. Only then it will be valid, and CDSU or Ayush or any review committee will approve. And all this data of your particularly protocol will be reviewed by the even the while marketing, while giving approval for marketing, or if you sell your product, the buyer will go through all this data, and your clinical trial protocol will be a very important document for them to take a call on whether your company or you have been very systematic and scientific in your approach. So ethics heading all this section is also extremely important. Next slide, please. So then comes quality control and quality assurance. Yeah, quality control and quality assurance. So here you must, for a small trial, let's say about 50 patients, 30 patients, even then it's better to have a third party visit. If you are an investigator, ask someone to come and review based on the protocol whether the case report forms. That is why I was asking annexures. Atrimed has asked you to attach some annexures, isn't it? That is informed consent form, which will be translated. I missed out that point. It should be translated into the local language, and that has to be taken by the patient. And you must attach a blank informed consent form, which has all the headings as per the requirement. So the monitoring process is when somebody comes and sees, have you taken the consent form? Some people take the consent after they start the treatment, which is wrong. So take the consent form, it will be dated, and the first drug will be dispensed, a case report form on which you record the data of the patient, blood pressure, blood glucose. Or that date should be after the date of consent. So that will be considered while monitoring, and that is called as quality control. And quality assurance is, and third party will come and see whether you have done the trial well, whether it has been monitored well. So both whether your quality control process is in place and whether you have done the study properly. So quality assurance department will be there with the company. If you are doing a trial as a doctor, still you can, after maybe 10 patients, ask someone to come and see the data, request the IEC to come and see. So like that, and document it. That can be done. So protocol will mention how you are going to do the quality control and quality assurance. Since this is a competition, I am giving you a tip that those who take care, some people have left, but if you take care of all these things in place, then probably your scores will be higher. When you write about what is a quality control, what is a quality assurance, even if it's a small number of patients, you want to doubly ensure that what you have done is correct, and that you write in your protocol. That will be very much appreciated. And then talk about financing and insurance. Why insurance is important? So generally in a clinical trial protocol, we mention about insurance because if something happens to the patient, and now in the CDSCO guidelines mention that you have to give a compensation to the patients, and there's a formula to calculate that, which ethics committee will calculate. If there is death, how much we have to pay. If there is serious hospitalization, then the person who funds the clinical trial has to pay, and that should be mentioned in the protocol. Sometimes, mostly they say whatever expenses are incurred in management of this patient will be borne by the sponsor. That is about financing, and the funding will come from so and so CRO. And what happens in this case? And insurance we buy so that when if anything happens, that insurance company will take care of the patient. Second insurance is for the investigator. If the investigator has followed the protocol and still something has gone wrong, it is not his mistake. So that therefore the sponsor buys an insurance for the investigator and sometimes for the institution also, so that they do not suffer any financial liabilities. Then we have the publication policy. Many times what happens is in academic institution, we are concerned about the publication because of the research we have done. So there may be some understanding between the sponsor and the investigator that this paper will be published together. You will get co-authorship. If you see Atrimed guideline, I saw that something related to intellectual property rights, that also can be mentioned here, though it doesn't come under GCP guidelines, but you can mention about this patent policy, IPR protection, all that you can write here and then do a protocol. So that is about an overview of protocol. It takes time, and thank you for patient listening. And I'm sure most of you will do very well, whether you get the award or not. At least learn how to draft a clinical trial protocol. Participating in clinical research will make your clinical practice very good, because you will develop analytical skills and you will start experimenting on your own treatment, and you will select what we call P drugs, what you feel is better. So I think it's very essential to build up a scientific temperament. So that is for today. And if you have any doubts, I'll be happy to answer. If I don't know, I will tell you, I can refer and come back. Thank you very much. Thank you, Atrimed, for giving me this opportunity, and thanks participants for participating. Good evening, Dr. Damle.
Can you just switch on your video? People want to see your face. Who is this brilliant lady who's speaking us, who has given us such a good lecture? We will go ahead with the question and answer session.
Yeah.
After that, I will talk to you. Yeah. People can type in the questions in the box.
Thank you. I'm sorry, in between, because of the net connectivity, we lost you.
Doesn't matter. Yeah. Doesn't matter, ma'am. Doesn't matter. If there are any questions, can people just put it on the chat box?
Can we orally ask the questions? If it is.
There's one question for clinical trial for medical devices. Was it answered already, or it's a new question? Ma'am, there is a question. How do we maintain clinical trial at OPD level? Any particular sample size? I mean, madam can answer, but sample size is depending on the clinic. I mean, the disease and the incidence of the disease, prevalence of the disease. It is not based on OPD or IP. Anyway, let me see whether we can hear from Dr. Pratibha. Okay, Dr. Pratibha is rejoining now, so please be patient. She will join back, and she got disconnected for some reason. Once she gets back, then she will answer the question. Dr. Ji, there are two questions on the chat box. Can you open the chat box and answer?
Can you hear me?
Yeah, I can hear you.
Can you hear me? Yeah,
yes,
chat box. Yeah, can you read? Yeah, I'm having some difficulty.
Okay. How do we maintain clinical trial at OPD level? Any particular sample size?
Okay. You answered that. Yeah, it doesn't depend on the setting, OPD or inpatient. It depends on the disease. It depends on the required, what phase, if you take about new chemical entity, and so that is a requirement. Which disease, what is its prevalence, and what you are looking at depends on your objectives. It doesn't depend on the setting. You can do a trial in OPD.
Next question is, throw some light on nano form of drugs. Want to understand whether it reduces drug effect on untargeted sites.
Yes, to some extent, yes, because even I was exploring it. Nano technology and nano-sized particles do have benefits, and particularly if you target them in cancer therapy, nano medicines have been effective because there is a specific expression of certain antigens there. But the drawback of nano is this: that safety. We are not able to get a targeted therapy in other diseases. Cancer, it has been to some extent, it has worked. But if it is not targeted, there is a possibility that it will have undesirable effects so far. But I have not worked on that, so my knowledge is this much. I explored for some diseases.
Ma'am,
Am I audible?
yes, ma'am.
Yeah.
Ma'am, what are the toxicity levels? Are they more than the other drugs comparatively, other active ingredients, or are they less?
Toxicity of nanoparticles.
Yes, ma'am.
I'm not sure, but they easily penetrate the tissues, no, because of their size. So that probability of organ toxicity is supposed to be higher. So what levels in the blood? Logically, their distribution is wider. If you take pharmacokinetics, their distribution will be much higher because they are nano-sized. That is what is predicted in some of the articles. But in cancer, it has been, yes. So levels, blood levels, if you are talking, I'm not sure. If you, I mean, you should refer to some particular compounds and
Yes.
I mean particular diseases and their treatment. Then maybe we can.
Like, ma'am, basically, I just wanted to, I've heard that they are more toxic. Maybe I wanted to just discuss with you whether the reason for that is because of the higher penetration they have into the blood because of their smaller form.
What I have read.
If the effect is good, the toxicity levels even in the minutest form will be higher. Is that how it?
Yes, that is what I have read. Yes.
Okay.
And that is logical also because they are nano sized and you know where the distribution and of course the quantity required will be very less because of their size also, and the dose also may be less.
Yes,
but this is what I have read.
and
Can I some chip in?
sure,
Please.
Yeah. Dr. Javid, see, toxicity and efficacy are two related things. Now, nanoparticles are used because for better absorption, whether through skin or through gut. Now, there is an optimal level of blood concentration for efficacy for every drug. If the and there at some level, all the drugs have to be toxic. There are no drugs which are not toxic. So now you have to see what is the level, what is the concentration you require for efficacy, and what is the concentration which can lead to toxicity. So it is a relative thing. If the toxicity is at much lesser than efficacy concentration, then that will lead to toxicity. If the toxicity is much higher than the efficacy concentration, then it is unlikely to lead to toxicity. So that's how you have to, it is a relative term. Toxicity is not an absolute value for any of a group of molecules like nano.
Okay.
So you need to
Sir,
investigate. You have to study. It cannot be generalized. You can explore.
yes, sir. So would the antidote be of some help while suppose we want to explore a drug, maybe we take a metallic drug or any ingredient or maybe curcumin may not be relevant here. But how about if we know there is a slight toxicity in that nano form of the drug, would there be a good role of antidote helping along with the drug if given along with the drug? If the antidote is already separately studied and use of antidote in that.
You can add on. Yeah, rather than antidote, if you can define its toxicity profile, Like which, see, just because it's a nanoparticle, it may or may not cause toxicity. But there are some hepatotoxic drugs, there are some causing renal toxicity. Maybe you can combine with some protective drugs. In Ayurveda, you have some hepatoprotective, for example. Maybe you can use them as preventive. I don't know, I'm not sure. Means I don't think we can add an antidote along with that, but you can protect some organs using, maybe you should explore that.
Sure,
I
ma'am.
think
Thank you
Dr.
so much.
Chikesh can add on if
Yeah, see,
anything is.
so it's again a very kind of difficult to answer question because antidote, whether antidote neutralizes the toxicity, the drug. If it neutralizes the drug, it will reduce the efficacy also. If that is the case, you
Yes.
can give a lesser quantity instead of adding an antidote. But if the antidote activity you're talking about is off-target, activity, then maybe yes. But then it's a lot of work to find out and
Yeah.
to give an answer, and it cannot be generalized at all. It's very, very specific to one molecule and very specific to toxicity and very specific to off-target activity. So it's very difficult to answer. But then idea is good. You can work on that.
I think you should targeting with nanoparticles would be better than using antidote or any other combinations. In my opinion,
Yes,
that
ma'am.
sounds little better. Yeah.
I
Any other
think
questions?
that makes sense.
Yeah.
Sure, ma'am. Thank you so much. Thank you, sir.
Prithvi, do you see any questions there in YouTube? Because there are many people watching on YouTube. If there are questions, can you just come back to me?
We do not have any questions posted in the YouTube live chat section.
Okay, in that case, anybody who wants to ask a question, I'll give you last chance. You can raise your hand, or you can just type it in the chat box, or you can switch on the unmute yourself and ask. Even later, if you can send a mail, we can forward it to Dr. Pratibha. She will be more than willing to answer your questions.
Yes, definitely.
I think no more
Yeah.
questions. So it's about time
Yes.
for me to give vote of thanks. Like anyway, these sessions went very well. We had a
Okay.
wonderful input from Dr. Lata in the morning about some ideas. I think many of those ideas were kind of eye openers. What we can do to find out the fact, and as Dr. Pratibha said, the outcome is not guessed in advance. Outcome is observed, and you just say yes, this is what happened, or no, this did not happen. So later we had some issue with Dr. Shenoy. He was summoned by some higher authorities for some immediate attention, so he could not join immediately. So there was a little bit of disruption. We are sorry about it. But however, he came and he gave a wonderful lecture about statistics and many other aspects. And lastly, Dr. Pratibha was brilliant. You can see how well she has grasped the subject and what kind of a hold she has over the subject and experience. Her talk spoke about it, and it is evident from interactions we had all this time. Thank you very much, Dr. Pratibha. It is
Thank you.
always pleasure listening to you, and Dr. Pratibha will be involved with the assessing your protocols, etc. And I'm sure she will also help you to improve your protocols to the level that the study can be published in a peer-reviewed journal with good impact factor. So
Yes.
we will work towards that, and it's not one day's affair or 1 year's affair. We will keep doing it, and one day we'll create a huge data about traditional medicine. And I conclude by saying we don't do research to prove something. We do research to observe and see whether something is true or something is false. Our hypothesis is true or hypothesis is false. Please submit your application with that spirit, and whatever is the outcome for that particular setting for those particular observations. That's how it is seen in clinical pharmacology. Thank you very much, one and all, and it's about time to disperse, and we'll meet again. All these lectures will be available on YouTube Atrimed channel. You can always listen to them, And I will, you can send any query to pmd@atrimed.com. I will just type it at the chat box. Please note it down. Alternatively, you can call us on 080-411-23727. We Atrimed is present on Facebook. We are present in Instagram. We are present in LinkedIn. You can connect us through that as well. Since you are all net savvy individuals, social media, savvy individuals. So thank you very much, Dr. Pratibha, once again, and let's call it a day. Thank you very much.
Thank you. Bye.